Peace be upon you all, and welcome to a new exciting session in our Seeing God in Biology series with Dr. Fuz Rana. Fuz, how are you doing today?
Good, Ahmed. How about you?
All is well, and I'm pretty much excited about the topic of today because I have always wondered why do we have those specific 20 amino acids building our bodies? Is there a reason for that? And did biochemists figure it out?
Yes. Well, this is actually a really fascinating and an important question, at least for biochemists. It is because when we catalog the number of amino acids that have been discovered, it's on the order of about 500 or so. And so that really begs the question: why do we see only 20 amino acids that are encoded in the genetic code that are used to build proteins? Why only these 20 amino acids? Is there something special about them? And biochemists are well on their way to answering that question in a way that's remarkable, that I think really highlights the creator's handiwork that we see in biology.
It also draws my attention that everybody's talking about the 20 amino acids as if it's like a given. So very seldom do and I think the audience will also agree, listen to science explainers talking about this topic. So they just say, and the genetic code will just translate to one of the 20. So I think many of them might have the impression that those are all 20 out there, but you're telling us that up till now, like 500 of them have been discovered already.
Yes, yes. And many of these in principle could have been incorporated into proteins if you are viewing life in evolutionary terms through the chemical evolutionary process that some people claim explains the origin of life. These other amino acids in principle could have been incorporated, into biological systems. And the set of amino acids is universal in nature. Every organism uses the same set of 20 amino acids. And so the question is this just an accident of evolution, or is there some deep-seated reasons for why these 20 amino acids are actually found in proteins?
But even if it's an accident, wouldn't it have been, like if we use rational thinking and think that this thing is just by accident, maybe it would have been 20 in humans, other 20 in I don't know, chicken, other 20 in, I don't know, insects and in plants, etc. Because if this is by chance, and maybe the translation system would produce different synonyms for the same codon, right? There is nothing
Yeah,
that mandates this.
that's one way to think about this. Now, the argument would be that this set of 20 was instantiated in the last universal common ancestor, and since from an evolutionary perspective all life derives from it, that instantiation has persisted. But to your point, as different branches of the evolutionary tree spawn, why wouldn't there be evolutionary processes that would alter that canonical set? Why wouldn't we see differing amino acids in differing organisms? And the fact of the matter is, we see this universal set. And so again, was this just an accident that was imprinted on Luca, or is there a rationale for this selection of 20 amino acids? And if indeed there's a rationale, that then really points us in the direction that this set of 20 amino acids was selected by
Yeah.
a creator for very specific reasons.
Yeah, because correct me if I'm wrong. To have different amino acids, all what we will need if this was by accident is a good luck accident between the ribosome and the transfer or the transport RNA. So this one will seek a translation for that codon, and the other one will just bring a different amino acid attached to it,
Yep.
and it has so it is not very difficult, given the amount of ridiculously, actually impossibly slim probabilities that evolutionists talk about, that if this was by accident, maybe this one extra accident could have happened through the
Yeah,
very long evolutionary history of life. Yeah.
yeah, that's exactly right. There's plenty of places where different amino acids could have been brought into play, and that the amino acid set from organism to organism would in effect be different, or at least be variable to some degree.
Okay, so since I'm very interested in the why question, let's go through your presentation and see if biochemistry would actually answer the why question. Because, as we've said before, if why questions have answers consistently, and why questions are typically seeking a purpose, then there must be a purpose behind all of that, and that can only be the creator, right or not?
That's how I would see it. Yes. There's
Great.
something profound about asking and answering the why questions. Yeah. So
Yeah.
let's get started. So this is just to
go.
set the stage. This is a cartoon of a protein, and in the top portion of the figure, you see a string of beads, and that's representing essentially the protein chain that is formed by linking together amino acids as building blocks. So each of those beads represents one of the 20 amino acids that is brought together in a chain-like manner. And then the backbone of that chain can fold into typically two distinct, what are called secondary structures, that are either alpha helices that are shown on the right, or what are called beta pleated sheets on the left. And then these regions of the protein chain that are folded can interact with other regions to form a three-dimensional structure. And the three-dimensional structure of the protein is really critical because it's what determines ultimately its function. And to first approximation, it's more complicated than that, but to first approximation, the amino acid sequence is actually going to determine the folding patterns of the protein chains. And so amino acid sequence is a really important characteristic of proteins. And then once you have a folded chain, it can interact with other folded chains to form, a protein complex, which is oftentimes referred to as the quaternary structure of a protein. And
Wow.
so the next slide shows, let's see here. The next slide shows, there we go. The chemical structure of an amino acid. Like I said, again, these are the building blocks for proteins. And so it all begins with a central carbon atom that's right here in the middle of the figure. And bonded to that central carbon atom is a hydrogen atom, is something called an R group, or a side chain. And then there's a carboxylic group, and then a amino group. And that's why it's called an amino acid, because it is a carboxylic acid with an amino group. And we can spend...
amino groups are characterized by the nitrogen, right?
Yes, the nitrogen with two hydrogens bound to it. And we could spend another half an hour dissecting the chemical nuances of amino acids. We're going to avoid that. But where we get the distinct flavors of amino acids is with respect to the R groups. The different R groups are ultimately what determine the chemical identity of the 20 amino acids. And so this is a diagram that probably is a little difficult to see, but it shows the 20 amino acids, where again, they all share in effect that same core structure, an amino group and a carboxylic acid group and a hydrogen, all bound to a central carbon atom, where the R group is different. And just simple inspection of the R groups shows that these R groups differ in terms of size and in terms of physical and chemical properties. So, for example, glycine and alanine have very small R groups, which is actually critical for the role that they play in proteins. Some R groups are what are called nucleophilic, meaning that they will attack other atoms, so that they have very important chemistry. And these particular groups tend to bind things like phosphate groups or or things like that. Then you have some R groups that are water soluble, some that are water insoluble. Some R groups have a negative charge, some have a positive charge, some are acidic, some are basic, some are aromatic. And so there's a wide collection of chemical and physical properties and sizes and chemical and physical properties and sizes that are represented by the R group. And biochemists love to study the role that different R groups play in protein structures. It's a common exam question when you're in graduate school, that if you won the lottery and you could replace, 5 amino acids in this particular protein structure, which 5 would you replace and why would you replace them? What would you think you would learn, by doing those experiments? So biochemists are really interested in those, those kinds of questions. Now, it turns out that,
yeah,
go
okay.
ahead.
I was just going to ask this question. So each separate amino acid, it looks like they look like each other, but then this R attached to them is what makes the difference. But how do they get stitched to each other to form this chain of beads that you showed us?
Yeah, and this is what's shown in this slide. Great question. And what you see here is 2 amino acids that are distinct, where one is represented by R1 and R2. These are just 2 different R groups. And the carboxylic acid of one will react with the amino group of the other amino acid in what's called a condensation reaction, where the element of water is removed from those 2 molecules. And as a result, a chemical bond forms between the carboxylic acid and the amino group. And that chemical bond is called a peptide bond, or it is also referred to as an amide linkage or an amide bond. And this is as we'll come to find out in a few minutes, a very unusual chemical bond. And that unusual nature of that bond actually plays a critical role in establishing the properties that are needed for proteins to actually form 3-dimensional structures. So we'll get into that in a minute. But
so
really,
you mean there is even an answer not only to why the 20 amino acids, but also to why amino acids altogether? Because if there is no peptide bond, then it might be a different kind of bond. If it's not an amino acid in the first place, something else maybe.
Yes, exactly. So and so we'll get into that here, maybe in more detail than anybody really wants to know. But it's, to me, the chemistry is absolutely fascinating. So this then leads us to the question that we're talking about today: is there something special about this set of 20 amino acids? Well, one way that we can answer this question is by varying the chemical structures of amino acids and see what happens. And so, for example, the first question is, why not an alpha hydroxy acid? And so an alpha hydroxy acid is an analog to an amino acid, where instead of the amino group, it's an OH group. Now, the reason why this is so significant is because if you remember the Stanley Miller experiment, where he generated amino acids trying to simulate the conditions of the early Earth.
That experiment, he fired an electric charge, I guess, through a flask that had nitrogen and other typically existing gases, and then the nitrogen would condense with the carbon and the hydrogen and form those, and he would see if he would get any amino acids, on their own or not, right?
That's exactly right. And it turns out that in that experiment, he did form some amino acids, but he formed many more alpha hydroxy acids. And so on the early Earth, if you think again in terms of chemical evolution, there would have been a preponderance of alpha hydroxy acids. They should have actually been used to build quote unquote proteins, not amino acids, because they're easier to make and they would have been more abundant. And in fact,
I see.
And in fact, just as again, amino acids can, whoops, let's see here. Yeah, just as amino acids can react together to form, again, a peptide bond, alpha hydroxy acids can react to form essentially the equivalent or the analog of a peptide bond or an amide bond, and this would be an ester linkage. And so you could form polyesters from alpha hydroxy acids. So instead of polyamides, why not polyesters is really the question.
Wow, we would have had plastic instead of meat.
Yes, exactly. But here's a slide showing a carboxylic acid and an alcohol, which would be the OH group, an organic compound with an OH group, and they react to form an ester. And again, this is an analog to the reaction that takes place to form the protein chain, which is a carboxylic acid reacting with an amine to form an amide linkage. So in other words, this is as close of a chemical analog to an amino acid that you can get as an alpha hydroxy acid. And in fact, this is a chemical reaction showing in that top left-hand corner a chemical compound called lactate. And this is the alpha hydroxy analog to an amino acid called alanine. And it can react to form, through a condensation reaction, to form polylactic acid. And
so now the question is, what is the advantage of the polypeptide, the peptide bonds versus those kinds of bonds?
Yes, well, it turns out that polyesters have their linkage has only single bond character, whereas polyamide, the linkage has a double bond character. Now, this has profound ramifications. But one way to think about this is in terms of the flexibility. So let's say you have two boards, and each of those boards represents an amino acid, and you drive a single nail between those two boards. They're held together, but they can rotate.
Yeah.
But if you drive a second nail through the boards, they can't rotate anymore. And so a double bond would be equivalent to joining two molecules together using, again, two nails instead of a single nail. Now that ends up giving that polyamide or the protein rigidity in the backbone that allows it to fold, but then when it folds, it remains in a stable three-dimensional structure because of that rigidity, whereas polyesters are floppy. So when they fold, they're just going to they can't really adopt stable higher order structures, which again,
because all the components have all those degrees of freedom, then adding many components will reduce the overall mobility of it, but it will still be fluid.
Yes, that's exactly right.
Wow, that's a huge advantage for this kind of bond, obviously.
It is. Now, this is where things get, I think, even more interesting. Now, the reason why amides have this double bond character has to do with essentially two properties that are different in an amino acid versus an alpha hydroxy acid. One is that the nitrogen in that molecule, in that bond, in the amide bond, has an unshared pair of electrons that can attack essentially the carbon, causing the double bond and oxygen to kick upward, creating double bond character. And the reason, but now you can't do this with a polyester because of the difference in what's called the electronegativity of nitrogen and oxygen. Because electronegativity is a property that describes the attraction of an atom for electrons. Oxygen has the highest electronegativity except for fluorine. It's not going to give up its unshared electron pairs. Nitrogen is less electronegative, and so it'll do that. And as a consequence, it creates double bond character. And so it's very subtle differences in the chemistry that have profound implications,
Yeah.
making the amide linkage a very this is what makes the amide linkage a really unusual chemical bond that has the just right properties, as we're going to see, so that life is possible. So not only, again, does it create rigidity, which allows for the protein to fold in a stable three-dimensional structure, but the double bond character makes it less susceptible to hydrolysis, which is the reverse of the condensation reaction. And of course, if hydrolysis took place, the protein chain is going to break apart. So polyesters are much more likely to hydrolyze than polyamides are.
Wow.
Now, it also changes.
Hydrolyzing means it dissociates from each other after it has come together, right? Like
Yes.
dissolving in the water medium or whatever medium it is in.
So the reaction that joins the amino acids together is called a condensation reaction, where through that chemical reaction, you're removing the atoms that would constitute water. And then if you reverse that reaction and you add water back, you break the bond apart and you regenerate the amino acid and the carboxylic acid. So polyamides or peptides made up of amide linkages are much less susceptible to that breakdown, which again is very important. Now, something else, and this is getting into some detailed chemistry here, there's another difference too. The polyester bond, it has a tetrahedral geometry, whereas the amide bond has a planar geometry. And so, and that is, again, really significant. So, let's see here. So, here we go. Okay, sorry, I got ahead of myself. So in other words, what we see here is that the carbonyl and the amino group are in the same plane, okay? And so when you build a protein structure, you have kind of these molecular planes that are rigid, that are again being used to build the three-dimensional structure. But as a consequence of that planar geometry, you can either have two orientations of the hydrogen bound to the nitrogen and the oxygen bound to the carbon. They can be cis, where they're on the same side of that bond, or they can be on the opposite side. That's called trans. Can you see the difference?
Yeah, so the hydrogen and the CH3 switch their locations on the right versus the left.
Right. Now this again is really significant because the trans geometry is favored because of what are called steric effects. And that just simply refers to the fact that two chemical groups can't occupy the same location in space. Not very complicated. And so molecules are going to adopt geometries to minimize those collisions of chemical groups. And so the trans geometry minimizes steric hindrance and is the preferred geometry. Now, it turns out that the only way you can get an alpha helix to form or a beta pleated sheet to form is if you actually have the trans geometry. If you had the cis geometry, you can't get these higher order structures. And so not only do you get this planar structure that allows for, again, rigidity to the backbone, but it forces a trans geometry which allows for hydrogen bonding, which makes, again, secondary and tertiary structures in proteins possible. Now, there's something else that goes on. This is, again, getting a little bit complicated, but in addition to the amide linkage, you have another chemical bond that's part of the backbone chain that these are single bonds. So you have the amide linkage, which is double bonded, and then to each side of the amide linkage, you have a single bonded carbon, and those are free to rotate, and those rotation angles are referred to as phi and psi. Now, in principle, you could have that rotation angle adopting anything from 0 to 360 degrees. But it turns out because of the amide bond, that double bond character, and because of the trans geometry and other steric factors, those bond angles are restricted. And it turns out that those bond angles are the only ones that are possible are the ones that actually lead to the formation of an alpha helix and a beta pleated sheet. And so what you have going on here is this conspiracy, right? Where everything is working in the just right way to allow for this amide linkage to form higher order structures that make proteins possible. So here are two biophysicists who are marveling at this. One cannot but marvel at how factors, the steric interactions.
How many factors? I think the many is, yeah.
Steric interactions, hydrogen bonds, which provide the scaffolding for protein structures, the constraints placed by quantum chemistry on the relative lengths of hydrogen bonds and covalent bonds, the near planarity of the peptide bonds, and the key role played by water all reinforce and conspire with one another to place proteins in this novel phase of matter. And so this is, suggesting that this is no accident, right? That there's
those guys are not creationists, are they?
Oh no, they're not at all. But let's think about this for a minute, because again, if it wasn't for the unusual properties of the amide bond, proteins wouldn't exist, and in effect, you could argue that life simply wouldn't be possible. And so either we got very, very lucky, or there was someone that predetermined ahead of time that this is the way things were going to be, so that proteins could exist and so that life is possible. Because in effect, proteins are among the most important class of biomolecules, because they're carrying out all the different activities inside cells that make life possible. And so if it wasn't for proteins, again, you wouldn't have the chemical complexity needed for life.
Yeah, because for the audience, correct me if I'm wrong, proteins are the tools, they are the machines, they are the transport, they are the scaffolding, they are structure, they are carriers like hemoglobin. They're just those small robots doing everything.
Yes.
And you cannot have a machine or a tool unless it has a shape. And what you have just described is that all the properties, you have the amide that reduces the degree of freedom, and then you have the single hydrogen bonds that gives it flexibility to shape, but combined with other factors, it can only shape into the basic shapes that will cause, from the primary to the secondary to the tertiary to the quaternary structures that will eventually form the machines that we want. Other than this, we would have had chaos.
That's exactly right. That's exactly right. And so this is why you have to have amino acids instead of alpha hydroxy acids. Now, the other explanations are going to go much quicker, because we've laid some very important groundwork here in terms of understanding, again, some of the details of protein structure. So, that leads us to the next question then, and that is, why not?
Why those? Yeah.
Why not diacids and diamines? In fact, this is what industrial chemists use to make nylons, and nylons are, in a sense, synthetic analogs to proteins. They are polyamides made by, again, diacids and diamines. Well, you could build proteins that way, but you're now introducing several orders of magnitude greater complexity into the system than is necessary. Amino acids, where the amino group and the carboxylic acid group are all contained in a single molecule, that's the simplest arrangement. If you had diacids and diamines, then you would have to have 40 different molecules at minimum, where you would have to have a diacid and a diamine both with the same R groups in order to build proteins. So this just creates unnecessary chemical complexity. So amino acids are the simplest system that you could work with in order to build molecules like proteins.
Wow.
Now, the other question is, why not beta amino acids or gamma or delta or N-alkyl amino acids? These are actually formed in Miller-Urey type experiments and are believed to have been present on the early Earth. So why not alpha amino acids? Well, it turns out, long story short, that by putting that amino group in different positions along the chain will essentially make hydrogen bonding interactions impossible. That having both the carbon.
Can you explain what it means to be an alpha amino acid?
Yeah. So let's see here. Maybe we could go back to that. Let me see if I can do that. Go back to that very early slide. Sorry about this. Here we go. So this carbon is called the alpha carbon because it's the carbon that is adjacent to the carboxylic acid group. Now, you could have another carbon atom. Let's say if we inserted a carbon atom right here.
Where?
Between the two, another carbon atom between the two carbon atoms here.
All right. Okay. All right.
If we did that, then that would create a chain where we have 3 carbons in a row. Now, that would make that carbon the beta carbon, because it would be one removed away from the carbon closest to the carboxylic acid group. If we added another carbon atom in there, that would now be, let's see, alpha, beta, gamma, because we're 3 carbons away. Delta would be 4 carbons away. But it turns out that the only way you get the right orientation of the amino groups and the carboxylic acid groups in space to allow for hydrogen bonding interactions that would stabilize an alpha helix or a beta pleated sheet is if you actually have alpha amino acids.
Specifically.
Yes. Now, an N-alkyl amino acid would mean that you, instead of having the 2 hydrogens, you would have actually another type of functional group here, like that's bound to where the nitrogen is bound to a carbon, and in that would also disrupt the formation of secondary structures. So it's really only is only an alpha amino acid that actually will allow you to form proteins. So and then you have to have a hydrogen in that alpha position as well, because any other chemical group would occupy too much volume, and that would be disruptive to the formation of higher order secondary and tertiary structures.
Because again, in 3D we cannot have 2 molecules occupying the same space, so the hydrogen is so small.
Yes,
it's
exactly.
the smallest actually one can get, because it's just a single proton. So at the end of the day, it's freeing space for other important stuff, so that when you're forming the whole 3D structure, there is enough space to articulate it. Is that what's happening?
Yes, that's exactly what's going on.
And we're talking about the hydrogen on the opposite side of the carbon that has the R group on the other side. So the hydrogen that was up there.
Yes, exactly. Right. So in other words, every detail of the amino acid structure is absolutely critical to what it's functioning in proteins. There's no detail left to chance. Everything is perfectly designed, is perfectly planned out. There's a rationale for why it is this way. Now, this leads us to the final question, and that is, well, why these 20 R groups? Couldn't there be other R groups that are possible? And this leads to a really interesting set of studies done by a biochemist, where they created random sets of amino acids with different R groups.
Just a question: what do the R groups have? It's like it can be a metal, it can be something like sulfur, like phosphorus, phosphate.
Yes.
What can be there?
So let's take a look at this. This is a little bit difficult to see, but for example, like glycine would just have a hydrogen atom bound there. Alanine has a methyl group there. Let's see. Valine has a, let's see, this would be a carbon that is bound to two methyl groups, as an example. You have, let's see, an OH group, that would be serine, an SH group, that would be cysteine. You can have, let's see here, like an amide group, that would be, let's see, asparagine, if it's a single carbon atom joining the amide to the alpha carbon, and if it's two carbon atom chain or group, that would be glutamate. So that just gives you a sampling of some of the different types of R groups. So it is clearly a collection of different sizes, different water solubilities, different chemical properties that do play obviously important roles in not only protein structure but protein function. But the question is, could you have other R groups? Is this just an arbitrary set that turns out to be useful, or is there something more to it? And it turns out that a team of biochemists have done a series of studies where they have randomly varied R groups that we see in the protein in the canonical set, where they just again randomly added R groups that aren't necessarily occurring naturally in proteins, and they discovered that the set of 20 R groups has an optimal set of properties that vary uniformly across size, charge, and hydrophobicity.
Wow.
And so, in other words, you get with this set of R groups, you get full coverage of these three really critical parameters that influence the functionality of the R groups. And this is what the authors of the paper write. The amino acids found in proteins form a highly unusual set of 20 amino acids. A maximum of 0.03% random sets outperform the standard amino acid alphabet in two properties. So if you just use size and hydrophobicity or charge and hydrophobicity, you would have a very small number that are a little bit better than what we see in nature. But when you have the three properties, nothing that they could generate, it displays greater coverage. So there's something really very special, about that set of amino acids.
And what you mean by the properties is it's like I'm thinking like we're using those Legos or those wooden bricks that kids play with. So you will need the pieces that are quite small and the pieces that are quite big, and all the different sizes in between. So this would be the even distribution. And then for the hydrophobicity, you would need the R groups that like water so much, and those that hate water so much, and everything in between. And what they're searching for is, can there be a different set of 20 amino acids that would have better gradient for all three properties, charge, size, and hydrophobicity? And what he's saying here is, if you try to optimize two, you get only 0.03%.
Yeah.
That's 1 in 3 in 10,000 that are better in two properties. But if you want to optimize all three, there's nothing that beats what we have, the 20 that we have. It's completely
So the amino, so this is why these 20 amino acids are used. So in other words, when you see optimality, number one, that's right away is to me an indication of design, right?
Absolutely.
Now evolution can optimize things, but to have this global type of optimization is not what you would necessarily expect. And the fact that there's a rationale for every aspect of the amino acid structure, and the fact that there's really no alternatives that actually exist, suggests that amino acids are really special materials that form very special bonds that make proteins possible. And you can't really envision anything that would be an alternative to proteins, because the closest possible alternative would be a polyester. And again, we showed that that doesn't work. And so you really are left with this eerie set of just right circumstances that make life possible.
Uh,
If anything was even a little bit different in amino acid structure, life simply wouldn't exist. And so it really does look like things have been purposed, so that
wow.
life could exist.
This actually reminds me of something that we use in engineering, and when we try to solve complex equations, searching for a maximum point using an algorithm, and you have those trial and error algorithms where you would set the algorithm to start at a certain search point and then it will go and search, or you can give it different search points and then it will branch out and search for the solution. And the problem with these algorithms is that once the algorithm finds a maximum, if you're looking for a maximum, or a minimum, if you're looking for a minimum, it means that if it's looking for a minimum, for example, if it goes to the right, then the value goes up. If it goes to the left, then the values go up. So it knows this is a minimum. But the problem with those algorithms is that once it settles for a minimum, it can call it a day, because it has no way to know if there is a minimum that is better than this minimum, or a maximum that is better than this maximum. And the only way that you will find the maximum of maxima is either that you will do a crazy search for the whole possibility space, or that there is somebody who is intelligent, who sees the global picture, who would tell the algorithm actually where to start so that it will hit the maximum. And an unguided genetic algorithm, this is sometimes what they are called, or trial and error algorithm, will typically not find the absolute maximum or absolute minimum, because it has no clue where to find it. So I think this only, to me, is very positive evidence of guidance and design, essentially.
Yeah, that's right. And to build off of your point, evolution can find optima or minima, depending upon the what's being optimized, but it's unlikely to be a global optima or a global minima. It's going to be a local optima or a local minima. And what this seems to indicate is that the set of amino acids is truly a global optima. It's not a local optima.
Yeah, absolutely. This is why don't we really hear this all over science explainers? I'm really surprised that
Well,
something like this is not really taught in schools in chapter 1 about optimization of life or something.
Well, I mean, it's a really great question, and I think it goes back to a point that you made in a previous episode, and even at the beginning of this episode, which I think is such a profound point that it bears repeating over and over and over again.
Yeah.
And that is, very few biochemists actually ask the why question. Most of what biochemists are focusing on is the what and the how. Right. And so they describe, the structure and the chemical composition of proteins. They try to understand what the different amino acids are doing in proteins, and maybe what is the advantage or the role of that particular amino acid, characteristically speaking. That's what I learned as a graduate student. And and it's of course very interesting, but it's when you ask the why question, that's when you begin to see whether that really is putting you in a position of saying, do we really see design, or do we see what it would be characteristic of accident, of unguided, chance events? And what's very clear is when we start asking the why question, that suddenly, we begin to see results that are rather remarkable. And so very few science. So what I put together here is really based on probably 2 or 3 scientific papers that have been published over a span of 40 or 50 years, where these scientists had the temerity to ask the why question, right?
Yeah, and the amazing thing is that there is an answer to every why. Why proteins in the first place, not polyesters, for example? Why, which says why amino acids, and why this kind of amino acids, not the dye and etc. This specific structure of them. Why the alpha amino acids? Why those specific 20 R groups out of everything that we have, actually? And actually, there is one more question. I'm not sure if there is an answer to it yet or not, but why 20? Why not 22? Why not 20? Why not 18? Why not 19? So I see the experiments you're describing are asking why those specific 20, but then why 20 in the first place? Because the codons can have 64 alternatives, right? But those 64 alternatives, there is a conspiracy that they map to only 20.
Yeah, that's a great question. I don't know that I have a ready answer. I mean, it might be related to the what we call the degeneracy or the redundancy in the genetic code, which we'll talk about in a few episodes, that is also highly optimized. Or it could be that the researchers that we quoted from since that paper was published that we quoted from have done other studies, and one of them is they were operating from within an evolutionary framework, and what they did is they started just randomly combining amino acids together, starting with sets of 3 and then randomly varying those sets of 3s and then moving up to sets of 4, 5, 6, 7. And what they discovered is every time in their randomization they added an amino acid that was part of this set of 20, they suddenly saw that the properties of that set that would be critical for protein structure and function shot upward in terms of value, right? Those properties went upward. And it was like, as soon as you added that right one, boom, that set became, dramatically improved. And so I suspect that they noted that certain amino acids, when they would have that effect, when in some cases when the set was really small, in other cases when the set was medium size, in other cases when the set was larger. So it wasn't always consistent. So it could very well be that there's something magical about that number 20, that's where you stop getting that
extra advantage. Yeah.
But so I don't know. It doesn't feel like a real satisfying answer, right? But it seems to suggest that there may very well be something magical about that number 20. I just don't know at this point, what it is, or if anybody's even, asked that question, or if you ask that question, how you might even go about really answering it. But these are the types of questions that to me are absolutely fascinating, right? These why questions, right?
And I will very comfortably bet on it that if somebody really does the study about what is the optimal number of acids, and he will really do the study, an extra one to the ones that you have said, and you will find that probably if it goes to 21 or 22, the complexity overweighs the betterness of the properties, and this would also again prove to be a maximum of maxima in set size and other stuff. So and the interesting thing is that from our perspectives as people who believe that there is a creator, a grand mind behind all of this, we are really comfortable asking the why question and searching for the solution, and we are comfortable that the question will yield results if enough funding and patience is put behind it. But in a different worldview where this is all happening by chance and there is no grand plot or anything, then somebody might say, well, you don't need to spend money on this. Who said that anything is optimized anyway?
Yeah.
But over and over again, it proves that things are always optimized, and there is always a reason, and there is something for us to learn. So I tend to say this, I'm not sure if you'll agree with me, that believing in the validity of the why questions, and hence doing research rather than not doing research, means that a person who believes in a creator is more active and more proactive, even in learning and finding benefits that can actually be used in biomimicry and other things that you've taught us about, rather than somebody who believes in randomness, who would maybe in so many of these situations will be lazy not to go and do the quest because his worldview is that there is no point to all of this anyway.
Yeah, I would fully agree with that. That, so often I hear the charge that if you believe in creation or intelligent design, and you say that God did it, well, that just shuts down scientific inquiry. But I think your point is that's not the case.
It's completely the other way around.
That it begs the why question, and through the why question, we gain fundamental insight, in this case, into the structure of proteins that we otherwise wouldn't have gained, which now allows us to apply that knowledge for technology purposes, as an example. And so in many respects, it really is, the evolutionary paradigm, or the materialistic paradigm, maybe more appropriately, that I think actually can get in the way of scientific discovery, to your point. So I would agree with that, wholeheartedly. It makes people very mad who are held to a materialistic perspective when you make that point, but that's been my experience.
Yeah, absolutely agree. And I once I had a discussion with an evolutionary biologist, and I had this at the end of my presentation. I was writing this way: a belief in in natural selection acting upon random mutation is essentially causing a laziness to the scientific endeavor, and pushing the human intellect into maybe a kind of well, I use the word retardation, but I think, or disability of some sort. It's handicapping human intellect from finding the real answers that will really advance science.
Yeah.
So thank you, Fuz, very much for this very, very critical topic that is very seldom talked about. And I hope all the audience learned many things today. And please give us your questions in the live chat. And if you want to discuss more, then maybe even join us in the after show, and you'll find the link in the live chat. So see you on the other side. Thanks again, Fuz. And this was really fun. And we learned a lot from you today.
Thank you.
Thank you. Thank you very much. So to next episode, where I think we'll be talking about this central dogma of biology, right?
Yes. At last.
Okay. At last. Yeah. So I can't wait. Thank you, everybody, and peace be upon you all. And see you next session, God willing.